The aim of the following experiment is to screen Hsp104 libraries for the selection of variants that suppress the toxicity of the proteins associated with protein misfolding disorders. This is achieved by co-transforming Hsp104 variant libraries with a disease-associated substrate in yeast and co-expressing the two proteins. As a second step, the selected yeast are treated with 5-fluorotic acid or 5-FOA to counter-select for the Hsp104 plasmid. Next, the hits are sequenced by amplifying the mutagenized region by colony PCR for identification of the mutations in the Hsp104 gene. Ultimately, the suppression of toxicity of the disease-associated substrate can be evaluated to identify the Hsp104 variants that are most promising for further characterization.
The main advantage of this technique over existing methods like binding-based selections is that this technique allows for the selection of variants that suppress the toxicity of the disease-associated substrate.
This method can help answer key questions in the protein folding field by identifying the proteins that can reverse protein misfolding.
Though this method can provide insight into Hsp104 potentiation, it can also be applied to screening libraries of any protein for the toxicity suppression of any substrate that is toxic in yeast.
Generally, individuals new to this method will struggle because many colonies may need to be screened to find valid hits.
To screen for proteotoxicity suppression, begin by washing Hsp104 colonies off the plates with raffinose supplemented dropout media, a serological